look at the requirements you need online. But today I'll be introducing Dr. Bill Hayes. He is a professor from Loma Linda, and he's a professor of biology at Loma Linda. As you can see his title. And some interesting facts about him. He actually taught Dr. Nelson, Corbett, and Dr. Thornton. So we all wonder how old he really is. No, I'm joking.
You can blame me for all the problems you're getting from those guys.
It's all his fault. Another interesting fact, he grew up in Maryland. At a young age, he said he would like to catch snakes. You said your dad got you into that? And that's what kind of led into his career of pursuing snakes pretty much. He also taught at Southern from the years 1990 to 1995, and he's actually the reason you are all sitting here tonight. He is the creator of these EOG lectures. You can thank him.
You can blame me for that too.
And then I asked him what his favorite hobby was outside his career, and I think this is kind of in his career, but he said his favorite hobby is birding, right?
Yeah.
And his favorite sports team, he's an Atlanta Braves fan.
Big time.
So if anybody out here. Without further ado, I will present Dr. Bill Hayes.
Thank you. Hey, you guys are at a great program. How many of you are biology majors? How many of you are not? I don't know why you're here, but I'm glad that you are. And this is general enough that I think you'll be able to understand a couple of these slides. Try not to fall asleep because you don't know where in the talk those will be spaced out. Actually, you'll probably be able to understand a fair bit. So title, I changed it just a little bit, but how many of you heard me talk a year ago? Anybody here? Okay. Can you hear me okay? All right. All right, Dave. So we're going to talk about rattlesnakes and their venom. And just by way of outline, we're going to talk first about the snake, the biological roles of its venom, the way it uses venom, and the composition of the venom. I'm going to try to minimize that because it gets a little technical. I could literally talk all day on these because everybody likes snakes. I shouldn't say that. Everybody wants to hear about snakes, whether they like them or don't like them. They still want to hear about them. So, and I've done enough research. There's just a lot to share. Then we're going to talk about the human aspect of this equation, envenomation, symptoms, clinical symptoms, and the treatment of bite. And then we'll even get into something really odd. Venom's designed to kill. We all know that. But it can be used to save human lives, which is pretty extraordinary. Okay, so let's get into the snake and its venom. I don't know, you guys are young, you probably don't know who Gary Larson is, but he came up with these Far Side cartoons back in the 70s and 80s. Fabulous sense of humor. This particular cartoon captures well the fear and misunderstanding a lot of people have about venomous animals and how they use their venom, okay? And at the outset of my research as a graduate student at University of Wyoming, there was a lot that we knew about the venom in terms of its biochemistry, its toxicology, and pharmacology. We knew relatively little about how the venom was used by the snake in its ordinary life, okay? And so that's going to be a big emphasis on what we do here. Rattlesnakes have a very triangular head. The same is true of the copperheads that you have here in Tennessee and the cottonmouths that you have further west in Tennessee and south and east of here along the coastal plain. But they're triangular because they have fat cheeks full of venom and a pair of venom glands, one on each side. Those are connected by duct to the fangs. The fangs are like hypodermic needles. They're hollow. They have an opening at the end. The venom gets squirted out, okay? My pointer is not going to work, unfortunately, because the batteries are dead. So I apologize. I'm going to have to stay close to my laptop, and I don't have a pointer for you. But you'll see that the fangs are ordinarily covered by this membranous sheath, okay? And that has to be pushed upwards. It happens very readily. There's no muscle to move it upwards. But when they bite and penetrate, the sheath gets pushed upwards, and that actually displaces an internal membrane, which ordinarily blocks the venom from flowing out those fangs and makes it possible for it to flow. you'll see there's often a backup fang. You'll see it on the right or the right side of the snake, the left side of your image. Here's the fang sheath as it normally appears in A, and in B, you see how the sheath goes up as the fangs go in, okay? So, venom serves two natural roles in snakes. The first is going to be for predation, okay? Not only does it immobilize and kill the prey, but it also helps to relocate that prey item, and I'll describe that in a minute here. And it looks like it accelerates digestion, makes it happen more quickly in the stomach after they've bitten that prey item. And I want to point out some prey actually are highly immune to the venom and can survive a bite from an adult rattlesnake. Pretty extraordinary. The venom's also used for defense, and there's two things about defense that makes the venom effective. One, over time, many of these predators have learned to avoid the rattlesnake. I shouldn't say learn, they have genes that tell them, oh, don't mess with this, okay? So there has been change over time where they naturally recognize that's a venomous snake, and it gives the snake protection. But if they do go ahead and attack the snake, there's generally a very immediate, painful sensation that can deter the predator and allow the snake to live, okay? And I want to add that some predators can eat these rattlesnakes like you're a possum right here in Tennessee. They have no fear of the venom because they have proteins in their body that protects them, all right? So here are some of the predators we're talking about. Hawks, king snakes will eat rattlesnakes, coyotes. There's no shortage of predators, okay? Now, when the snake eats, and this is how the venom is most often used by the animal, they have a pretty extraordinary way of doing this, okay? They usually detect their prey with their eyes and their facial pits. That big hole that is right here, that's a facial pit that's used to detect body warmth, okay? It sees infrared wavelengths. They have no ears. They're typically just sitting in one place where there's rodent odors, waiting for the animal to come by, and they ambush it. They strike it, they inject the venom, they release the animal. Pretty weird. Now the animal runs off some distance and dies. That snake has to relocate that wounded animal. Often they remain immobile for a period of time because they don't want to attract the attention of this dangerous mouse that could come back and bite them in retaliation. And then they'll start flicking their tongue, and we call this chemosensory searching, strike-induced chemosensory searching, where they're following the chemical trail left behind. And their sense of smell is so extraordinary that, think about it, there's two odor trails, the mouse running in, the mouse running away, and they can tell the difference because the venom, when they bite it, instantaneously changes the odor of that rodent. There's a component in the venom that looks like it's associated with that change, okay? And they can successfully relocate that prey just flicking their tongue and following the chemical odors. When they do so, they swallow it whole, okay? So this entire sequence, which is really very impressive, clearly the successful strike and injection of venom is the most critical for success, all right? Here's that facial pit that I mentioned, and here's what a mouse would look to them on a moonless night or maybe down in a burrow. They can see even the front end of the mouse. I had a colleague who studied a congenitally blind snake. It had no eyes, but very functional facial pits. And that snake would not only strike mice accurately, but it would preferentially strike the front end of that mouse because it could see the warmer end of the mouse. Is that pretty amazing? Okay. This illustrates another predator-prey interaction here. This is the ground squirrel. The rattlesnake likes to eat the pups of the ground squirrel because the adults, it can eat them too, but a lot of them will survive a bite from even a full-grown rattlesnake. So the snake on the left is confronted by a ground squirrel that sticks its tail up in the air and waves it, and the squirrel is communicating to the snake. Na 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 na, I see you. I'm fast and you can't catch me. It's doing the same thing to the harmless gopher snake on the lower right, but there's a big difference in what the squirrel does. The squirrel, when it's confronting a rattlesnake, shoots warm blood up into its tail so that the snake can see it even better. That squirrel is communicating to the rattlesnake using a private communication channel. That rattlesnake, remember, can see that warmth in the tail, so it's a stronger visual stimulus. Amazing. all right? Again, I've mentioned how the venom instantaneously changes the odor of the prey. They can tell those two trails apart, and they almost always swallow the prey head first they recognize that mainly by the odor differences between the two ends, okay? So I wanted to know, considering the importance of this venom, how much is injected during a bite, and can the snakes control it? And when I first began my research, there was a published abstract that said they can control it, and there was a published study using a European viper that says they can't, okay? So my Ph.D. work was actually working with the rattlesnakes. Briefly, the bullet hypothesis says it's always a bullet size, it's always the same amount every time they inject. Venom metering hypothesis simply says they can control how much venom they deliver when they bite, and they make decisions about how much to inject. And we could consider other hypotheses. There may be some kinematic influences. Maybe the speed of the bite might influence something. Maybe the duration of the bite, more time for venom to flow. And I'm going to tell you that the second and third ones, good data support. Good data certainly support the second hypothesis, the venom metering hypothesis. And we'll look a little bit at that real quick. Before I go further, I want to ask you a question. Which is more dangerous, the baby snakes or the adult snakes? How many of you say it's the baby snakes? How many of you say it's the adult snakes? It's about 50-50. Actually, I think more of you say it's the adult snakes. If I asked an audience in California, more than half the hands would say it's the babies, and I would get maybe one or two hands go up to say it's the adults. We'll get back to this. We will get back to that. All right, let's consider how much venom the snakes use in predatory bites, okay? This might remind you of your cafeteria there, perhaps. But snakes dream of food. They're eager to eat. All right, so we see here snout vent length down below. On the left, small snakes. On the right, the big snakes. And the amount of venom that's injected into mice, small mice, medium, or large mice, corresponding to the size of the snake. Who are they injecting? Who is injecting more venom into mice, the big snakes or the little snakes? The big snakes. It's an exponential increase. As the snake gets bigger and bigger, the amount of venom exponentially increases. And that actually corresponds to the amount of venom that's available in the gland. Bear in mind that as the snake gets longer, the venom gland is going in 3 dimensions, okay? And so you get more and more venom contained there. And we can look at prey size, and you can see here that, see those lovely mice. So I just want to say that the small, if you look at the white bars, that's the medium rattlesnakes. They're injecting more venom into the large prey than into the small prey. And for the darker bars, more venom into the large prey. Those are the big snakes injecting more venom to the large than the small. Okay? So this suggests that the snakes are able to control the amount of venom they inject. When I look at the videos, frame by frame, or because they're digital, we call it field by field, we see no differences in the duration of the bite or other aspects of the strike. So I think they're not only making a decision to inject more venom into larger prey, which can live longer and run further after they've been envenomated, but I think that the snake is making the decision before it strikes, which is amazing, okay? It turns out venom metering, we have good evidence that occurs in other contexts. When they deal with different species of prey, I'm not going to get into that, like birds versus mice, different sizes of prey, different levels of hunger. I think there's a difference in predatory and defensive contexts. Even the level of defensiveness, when a snake is much more defensive, I'll show a little data, I think, on that. And duration of the bite. A longer bite does allow more venom to flow in. I'm not going to take the time to show you all the data sets to demonstrate that, but I will add that some people have told me, oh, come on, snakes can't make a decision. Their brains are too small to have this kind of understanding. But we know that cnidarians, what are cnidarians? Jellyfish, anemones, corals, cone snails, spiders, scorpions, ants, even much simpler animals are making decisions at some level about how much venom to inject. The work by spiders was done by Dave Nelson. Work on scorpions was done by a couple other students in my lab. Sometimes I wonder who's got the small brains, whether it's the animals or the humans. By the way, does that look like a former president of the U.S.? I don't know if you guys remember George Herbert Bush. Anyway, Let's talk about defensive bites, all right? So I think the evidence is clearly there that the snakes inject more venom into different prey species and sizes. For defensive bites, the best way we came up with, we could have had students and volunteers come in and let them get bit and measure something there, but we decided the gloves would be cheaper and less noisy and all the mess associated with that. So we just fill them with saline. We get them nice and warm to resemble a warm human limb, and we dangle them in front of those snakes and piss them off, and lo and behold, they bite. So we could use a simple enzyme assay to measure the amount of venom. And who is injecting more venom defensively here, the little snakes or the big snakes? The big snakes, clearly, okay? And there's just no question about it. This was work done with our rattlesnakes from California in orange and cottonmouths in black. And these are 3 consecutive bites, and you can see that by the time you get to the 3rd bite for the rattlesnake in orange, the amount of venom looks like it's starting to reduce, but you still get a lot of venom being injected, okay? That old adage, first person walking by on the trail wakes up the snake, the second person the snake strikes at and misses and hits the 3rd person. But if it bites again, every other bite, there's less venom. That's not necessarily the case. Look at the cottonmouth in black. The cottonmouth actually appears to give you less venom in the first bite, as if it's saying, back off. And if you don't get the message in the 2nd and 3rd bite, it may even give you more, okay? So there's a real species difference there that's significant. All right? So these are fairly simple experiments to do. Fairly simple. You guys could do something like this, where you have a snake that is offered a big mouse, and then a few weeks later, he's offered a small mouse, okay? And half the snakes get the mouse first, and then the little one, and then the big one, and the other half of the snakes get the big mouse first, and then the little mouse. And all you're doing is measuring the amount of venom. It's not necessarily simple, but overall, the experiment is something anybody could do, okay? So I want to say something about venom extractions. Yeah, they're a little dangerous. They're essential for research and anti-venom production. And I want to show you how the venom comes out of this lovely creature. So you're going to watch the fangs. You'll see the right and left fangs function very independently from each other. And you'll see that there goes the venom, big squirt. And now you'll see the right fang swing back and then come at it again. These have punctured a membrane. And now the left fang is going to do it again. We can also see in multiple pulses, we might see a long pulse followed by a short one, and then maybe a long one. It's very clear that the snakes have good control of how much venom they squeeze with their muscles out of the glands and through the fangs, okay? You like that? It's pretty cool, huh? Now watch this. Watch the tongue flop from the right side over to the left side as it launches another defensive strike at an inflated glove. Okay, and you see the fangs in action there. Okay, now don't get any ideas about what you can do playing with these snakes. That's filmed at high speed, so when you play back at slow speed, at normal speed, it's very slow. But that strike is actually about the speed of Muhammad Ali. Okay, and don't think you can dodge that so readily. Not all the students in my lab, by the way, love snakes. This is a kid from the U.S. Virgin Islands. Some of you knew Tony here, just probably the faculty in back, Tony Trim. All right, sometimes we get dry bites. This dude got real lucky there. That's what we call a fail. But let's consider protection, the possibility that wearing long clothing might interfere perhaps with the mechanics of the fangs and their movements. Could we get less venom injected when a snake is biting through this material? And the answer is yes. So in orange we have naked gloves, and in black we have gloves that are protected by denim material. And you'll see that whether the snake is small or large, there's about a 2/3 reduction in the amount of venom that's injected when you cover that glove with denim. So this suggests that when you're in natural snake habitat, if you're wearing long clothing, you may have some benefit, okay? You may have, if the snake does bite you, you may have a reduction in the severity of the bite. Okay? While we're dealing with defensive bites, let me add a really interesting and fun study here. We know that many animals that live on islands are unusually tame. You go to the Galapagos Islands, photograph birds like these blue-footed boobies, you can walk right up to them. Amazing tameness. We know this is true of many reptiles. This island tameness phenomenon has been known for centuries, more than a century. When it comes to rattlesnakes, we have some island, the word insular means island, we have some populations in Baja California where the snakes don't have rattles, or at least a large number of the rattlesnakes don't have rattles. And we think this is kind of evolved, we're talking about microevolution here, because there's a lack of predators, okay? And so what's happening is this basal segment of the rattle has this shoulder, it has a narrowing and then a shoulder, but the shoulder doesn't develop well, so that every time you add another segment, it just slips right off, okay? And so we would expect island rattlesnakes to be not so very defensive, okay? Now we have the Southern Pacific rattlesnake, which I have a lot of data on from California. That's California on the right. You'll see some islands offshore, the Channel Islands. One of those islands, Catalina Island, just off of Los Angeles. That gray area to the right of it is all Los Angeles. Catalina Island has a Southern Pacific rattlesnake. It's kind of midget in size compared to the mainland. And the rattle's perfectly normal, okay? And they use the rattle. And my students, when we were doing a little study with them, they said, these snakes are nasty. They're hot. They really are defensive. And I'm like, that shouldn't be the case. That sounds kofefi to me. I don't know if you know that word. So we did a little experiment where we had the snakes, I would pin them and pick them up, and we would record some behaviors. So you can compare 20 mainland snakes that we had in the lab, 10 snakes from the island. You'll see no significant difference between whether they rattle before they grasp, or rattle during the grasp, or rattle during venom extraction, or even cloacal gland discharge. But look at the difference in attempting to bite. A high percentage of these Catalina snakes, they would bite. And because they're doing this, it took more time to capture them. This was done while I was blind. I wasn't sure which snake source I was dealing with, whether it were mainland snakes or Catalina snakes. The amount of venom injected was like double by these snakes at any given size compared to the others, and the amount of venom per bite was also double. They basically had the same number of pulses during our defensive bites into the beaker, and here's just a look showing you that at any given length, even though the island snakes with the white dots are smaller overall in size, but any given length, you can compare the two and there's a significant difference. Those island snakes, which should have been more tame, are paradoxically nasty animals, very defensive. So of course we wanted to know why that might be the case. We looked at a number of options, including numbers of predators of birds, predatory birds. There's actually fewer on the island. We thought maybe migrating birds, birds of prey would cause this. That's not the case. We looked at mammal density, and these are the carnivores that eat other animals or prey on snakes, and omnivores. Catalina has few there, but it does have fairly high densities of the introduced cats and pigs, tons of pigs at one time, okay? So that might be part of the explanation, but then we also have these herbivorous ungulates like deer and buffalo and cattle and goats and sheep, and there's been tons and tons of them on this island that have actually denuded vegetation in large sections historically. Now things are looking better. But we've had all these animals running around. I'm going to call the ones on top predators and omnivores. The pigs will eat them. The ones down below are, I will call them antagonists, because they'll actually stomp and kill these animals, rattlesnakes. They're not trying to eat them, so they're not predators. But you'll see from the snake's perspective, here's some big creature there, and we think because humans introduced, these are, only the fox is native on this list. All these introduced animals in Catalina, in very high density, we think, has caused this change in the behavior of the rattlesnake, which kind of tells us that when we screw around with the environment and we're not thinking about what we're doing, in this case, we have a more dangerous animal. an animal that carries more risk when you're playing with it. And we'll talk about playing, we shouldn't do. All right, here's the question I got to earlier. Are babies more dangerous? And the answer is no. And the main reason has to do with the amount of venom available. Again, the big snake has much more. In fact, the baby's entire head may be smaller than the individual venom glands on the adult snake, okay? And the adults just have way, way more venom, all right? So I wondered where this myth might have come from. I went to old historical newspaper stories, and I found that before 1970, in the left column there, all the news stories from California and other parts of the US pretty much got the story right. In black means that it was correct. Then between 1970 to 1999, the middle bar there, the metal panel, in California, all the stories were wrong. Other parts of the US pretty much had them right. And then to the right, after about 2000, everybody's getting it wrong everywhere. That's the internet age, right? And so I think this myth probably started in some stupid place called California, like maybe around 1970, okay? And as embarrassing as that is for somebody who now lives in California. But I surveyed college students, and I think I have a sample from Southern Adventist University in the Southeast. But I surveyed college students, and the sample there, N equals, that's the number of freshman biology classes that I sampled. And you'll see that familiarity with this myth declined as I went east and to the north. Okay, so again, I think the stupid myth started out in California. Let me be clear. Who is more dangerous, the baby rattlesnakes or the adults? The adults, way, way more dangerous, okay? But you'd be surprised, 53% familiarity with that myth, and they buy it. People buy it. And there are people who, I mean, tons of data to support this. There was never a shred of evidence to suggest the babies were more dangerous. But there are people, maybe like some of you, who hear somebody like me get up and tell you what the facts are, and they don't believe it. They don't accept it, okay? And we need to think about the facts that we hear. It does help to do a little background search and look at the quality of the claims, okay? Let's talk about venom composition. It's viscous, usually yellowish liquid. There's a particular component responsible for that. There's many enzymes within an individual snake's venom. In rattlesnakes, that's often as many as 100 in a single specimen's venom, different proteins, different enzymes that are toxic and doing the damage. We have two basic types. The proteolytic, which simply means it's digestive. It kills by causing you to bleed out, basically. That is less toxic. Most of our rattlesnakes have that. The neurotoxic tends to kill by paralysis. It causes your muscles to stop working, including the muscles that help you breathe, okay? And that's what we typically have in cobras and kraits and mambas and all those exotic snakes that are long and slender, but our vipers that are fat with skinny necks and triangular heads, including our rattlesnakes, your cottonmouth. Most of them have the proteolytic venom, but a few of them have some neurotoxin in their venom, sometimes quite a bit, okay? We tend to see an ontogenetic change in the snake venom. The young snakes tend to be more toxic to kill the lizards, not necessarily neurotoxic, but just overall more toxic. And then as the snake gets bigger and bigger, now it has a lot more venom available, and usually it's digesting, consuming really large, bulky rodents that are difficult to digest, so they change the venom to become more proteolytic or digestive. Does this make sense to you? Okay, and even the neurotoxic species, it looks like the babies and the adults don't have much change. We call that pedomorphosis, the retention of the juvenile trait, more toxic, into the adult state, okay? So that just gives you an idea of what's going on there. At Loma Linda, we're able to explore differences. So using this wonderful technology where we have a liquid chromatography system on the left, and I'm looking at two slides side by side. So that's the HPLC system. We run our samples through that. I'm not going to describe how that works, but I will tell you that the fluid that comes through the column moves from the earliest stuff that comes out is on the left, and then as it comes out later through that column, it's on the right. And we will see different components, venom components, come out during the 2-hour or so run that we have, okay? And in different colors, those are different toxin families, and it's cool stuff. I'm gonna point out real quickly that these over here that are green, those are myotoxins. Those are myotoxins that damage your muscles, and they make your muscles crawl under your skin. Now, I don't know what that feels like, because I'm pretty careful, but there are people who know all about that. In red, that's a neurotoxin called Mojave toxin that's present in some rattlesnakes, and it's really one toxin, but it separates into two components under these conditions. And then over here, these are snake venom metalloproteases that are very digestive. So I'm going to point those three out in a slide soon to come. In terms of variation, this is a single species of southern Pacific rattlesnake in Southern California, and you can see how variable the venom is from location to location. And if you look, for example, here, some snakes have a lot of snake venom metalloproteinases, some don't. We have a population that has Mojave toxin, the neurotoxin, elsewhere it doesn't. And usually we see lots of myotoxin, but C up there in the white rectangle has no myotoxin to speak of. So there's a lot going on there. Other rattlesnakes that we've looked at, because we've had so many in Southern California to examine, they don't show anywhere near this level of variation. And we could continue talking about this. I've already discussed that, so I don't want to get back to that. A lot of this ontogenetic variation that we see corresponds to the diet. This simply shows how the Southern Pacific rattlesnake shifts between lizards, mostly eating lizards in blue when they're young, and then as they get bigger and bigger, they shift over to mammals, okay? Let's talk about super toxic snakes. There were some newspaper stories coming out in the early 2000s, around 2007, 8, and 9, I think, where newspapers in San Diego, California, Arizona, and Colorado were all claiming, doctors were claiming that we are seeing super toxic snakes evolve very quickly out of nowhere, causing really bad snakebite symptoms. Total nonsense, absolute nonsense. The venoms can't change that fast, number one, and there's other issues involved there. And people are generally saying we have some changes in the severity of bites. They attribute it to the snake's venom composition. It could have been just the size of the snake. If you're in a drought, for example, you don't get much reproduction. So a lot of the bites aren't being caused by babies, which we see a lot of in the fall. Aaron Corbett shows in some of his research. If you don't have many babies there, more of the bites are being caused by adults, which would cause more severe bites. There's also changes in humans. Humans are becoming more obese. We're becoming more allergic, hypersensitivity. I can't think. What is the name of the condition where you have to take a thing to help you breathe? Asthma, yeah, asthma has been really increasing, sorry. And so you would expect more severe bites. But the biggest problem here is that there's no data whatsoever to show there's been a change. And even if you had it, you had a change in the antivenom in 2000. Everything before 2000 was one kind of antivenom, everything thereafter a different. And so it's just a sensationalistic story that newspapers are telling. And when a colleague and I published some of this stuff, it all shut up. It just stopped. Talk about super stupid humans. Maybe instead of super toxic snakes, we have this going on instead, okay? How about that one? Yeah, you'd think natural selection would take its course here, wouldn't you? Okay, so let's move now into the humans and snakebite. In the US, we have probably 4,000 bites a year. In the old days, the number everybody said was 8,000, but it's hard to know exactly. More recent estimates are probably more accurate. Only about 5 to 10 deaths. So your odds of surviving are going to be close to 999 out of 1,000. But you might wish you were dead, because those bites are bad, okay? In the world, probably closer to 2 million bites, and probably close to 100,000 deaths a year, okay? And so we're talking about a serious health issue, At Loma Linda, we've been interested in what might cause the severity of a bite. Has Aaron Corbett talked about this recently? Did I hear all day? All day. Good job, Aaron. Tell more of your students, because I only saw 2 hands go up. Where are you, Aaron? Is Aaron here? He's what? He had to miss it. He's heard all this. Okay, so Aaron compiled these data. It took him a lot of work. 354 snakebite cases, 166 of them we knew that bit the patient, including bites from 7 different species of rattlesnakes in Southern California. Aren't those lovely? How many of you like snakes? You gotta be kidding me. How many of you hate snakes? Well, you suppose I could change that? Would you be at all open-minded? Because they're more scared of you than you are of them. I can tell you that when you get near them, their heart rate just starts racing. I've put electrodes on them. Their heart's going tum, 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 you know. You think yours is, theirs is too. All right, so Aaron scored 6 clinical symptoms from the pulmonary, cardiovascular, local wound, gastrointestinal system, hematological, neurological systems, okay? And so you add them all up, you get what we call a snakebite severity score with a maximum of 20, which means you're dead. Nobody got that high. 16 was our highest. I think that was a fatality. But again, when we look at the factors that statistically influence how severe a bite is, only 2 factors were significant, okay? So patient masses, so the young people, small patients, they had more severe bites. The big patients had less severe bites. Does that make sense? Big people, we can absorb all that toxin a little better. And then size of the snake. Here are the big snakes. That's the average line from the medium, average line for the small. This is only a subset of 100 snakes, but Aaron's data look identical when you get all 354 in there. It's pretty impressive, okay? So that's more important than the species. It's more important than the location of the bite, okay? And he actually gets the very same measures when he looks at the number of vials of antivenom used to treat the patient, or even the duration they stay in the hospital. Same relationships, okay? We can ask, where were the bites distributed? Upper versus lower limb. Statistically, we can identify which factors influence that. I could ask you to guess, but we need to hurry here because I know you have another meeting to go to. Turns out patient sex and illegitimate versus so patient sex. Males are more likely to be bit on the upper part because we're stupid, okay? Illegitimate bite is when you're messing with the animal. Legitimate is when you got bitten and didn't know it was there, okay? So if you're messing with it, you're more likely to be bit on the upper limb. How about the distal versus proximal? That's like beyond the wrist versus before the wrist or the ankle, okay? And it turns out the factors there are going to be snake size. The little ones generally bite you out here. The big ones get further up. And again, if you're messing with the snake, you're more likely to get bitten further up. Patient sex, patient age didn't really make a difference. And then lastly, what influenced whether you were interacting with the snake and got an illegitimate bite? What do you think? You can shout it out. What? Oh, alcohol. Heck yeah. Alcohol and men, okay? So, snake bites are associated with the 2 most dangerous chemicals in the world, testosterone and alcohol. Am I right? 2 most dangerous chemicals in the world. We can look at multiple bite cases. Unfortunately, all these patients that come in, we probably took all these variables. We should have taken IQ. But anyway, we did not. But here's a patient, a 44-year-old male gets bit in the left hand, and it's like, oh, and he gets bit in the right. I don't know what he was thinking. Here's an 8-year-old male, and a 4-year-old says, hey, I can do that too. And but anyway, the point here is that the first bite is often as severe as, I should say the second bite is often as severe as the first, okay? And that's simply backing up the data we saw earlier showing that by the time you get a third bite, it can be still fairly severe, okay? Now we can ask, snakebite severity is the same for all 7 species, but what about the clinical symptoms? We've got 6, remember. Maybe there's some differences among those 6, okay? So let's look at that. Now this looks complicated, but I'm gonna walk our way through this. So here are the clinical symptoms. If you're above 0, we standardize these. So 0 is the average for all 166 bite cases. If you're above the average, that means you got really bad P symptoms. That's pulmonary. Okay? And if you're below, like yellow, you got less than average hematological symptoms. And then this is what's in the venom, as I've shown you earlier. So let's walk our way through this. You'll see that these two species, the western diamondback and the red diamond, they've got a ton of the snake venom metalloprotease, the really digestive stuff in their venom, and that's what causes really high hematological bleeding problems. See the yellow bar way up? Bleeding problems. Come on, guys, I have 6th graders who can look at those figures and understand this, so follow me. Look at this. This is the southern Pacific rattlesnake. It's got really high neurological symptoms. Now, those neurological symptoms included the muscle fasciculations at the highest level. And those aren't necessarily neurotoxins which cause this, but they have a big load of these myotoxins in their venom. And that's what's responsible for the muscles going crazy. And then we have another example here. The Mojave rattlesnake has a lot of this neurotoxin called Mojave toxin. Now, it doesn't cause the muscle fasciculations, so we don't have very high neurotoxic scores. And the people who came up with the scoring probably should reconsider how they do neurotoxins. But look what they have. They have a ton of pulmonary, ton of cardiovascular, and a ton of gastrointestinal issues, okay? So the physician needs to be looking at those and saying, we got a problem here. Unfortunately, when it comes to a physician deciding how much antivenom to give, how many of you want to be a physician one day, an MD? A lot of hands. Okay. So if you want to know how much venom to give, you read the package insert. And the package insert says that if you see a lot of wound issues, like swelling and bleeding issues, that's when you give antivenom. But for this species, you don't get much wound or hematological bleeding issues. All the others, you get a lot more. And so a physician may undertreat a bite from this species unless they are aware that you get something clinically very different. Now, these data were collected in a hospital by a physician who was well-informed and who recognized, but elsewhere, a lot of physicians don't understand that. So this is helpful to understand the distinction or the relationship between the venom composition, or venome. Venome is the protein component of the venom, and the clinical syndrome, all right? So I've just described how we can infer syndrome from venome. I'm going to very quickly see if we can go the other direction, okay? So here's an interesting case. Neurotoxicity following envenomation in the sidewinder. Sidewinders have never been reported to have neurotoxic venom, okay? And this report came from, there's the range in red of the sidewinder. We have sampled venom from all over the US portion of the range, over 100 venom samples. And this is what we're looking for. There's the neurotoxin that would cause this neurotoxicity in that particular patient. Cerastes in this one below does not have that neurotoxin. So that's what we're looking for in all these venom samples. And we were initially told that this snakebite patient was bitten near Yuma. We went there, caught a sidewinder, and had normal venom. And then the author of the paper stopped emailing me because I asked him for a photo of it, and he wouldn't give it. I don't think he had the photo, but I suspected it was not a sidewinder. It was a Mojave rattlesnake instead. And then I discovered another author 2 years later that I could reach. And so I contacted her. She says, oh no, it's at another location in South. She gave me the exact location. We went there and caught 5 snakes in the general vicinity. All those had normal venom, and she managed to track down the photo that she had shared with the professional herpetologist, and it's a sidewinder, clearly a sidewinder. So we had this really bad neurotoxic bite there, but yet we can't find venom within the population. We thought maybe there was an isolated range where some of the snakes had different venom, like we see in the Mojave or the Southern Pacific rattlesnake. Couldn't find it. So we were unable to find a good explanation for why that patient had really weird symptoms, okay? And then we have the Mojave rattlesnake. The Mojave rattlesnake is very neurotoxic throughout its range, has that Mojave toxin, except 2 areas, south central Arizona and down in Mexico. And a colleague of mine in California had a patient bitten in California that had really strange symptoms. They were not neurotoxicity. They were more this digestive, this proteolytic venom. And he had photograph of that snake They chopped off the head, and it's clear a Mojave rattlesnake. So we went out there and sampled that region, and we had, I think, 4, maybe 5 venom samples. They're all very neurotoxic. We couldn't find a sample that was absent. So here we have 2 cases with really strange symptoms, and we don't have a good way to explain it because those local populations seem typical of the species, okay? And again, that's all we were looking for, those 2 peaks, okay? It's easy to identify whether they have this neurotoxin, okay? So we can ask why we got these bad snakes. Maybe it's misidentification of the snake, but in these 2 cases, the snake was properly identified. It could be maybe this, some humans react more with these symptoms than others. Maybe there's some extreme there that we observed. Maybe that snake's venom that bit the patient truly was unique. We just don't know the answer there. And this is important because we want to make sure that patient gets the right treatment, okay? We want to make sure they're getting sufficient antivenom. We want to make sure that other supporting care is proper. And we're also interested in how that venom evolves, how it changes locally, regionally, what factors. I mean, I could give you research showing how we think diet doesn't have much influence on rattlesnake venom. Other species of vipers in the world, it does, but our rattlesnakes, the diet doesn't seem to have much effect. We're exploring much of that right now. I'm going to finish this a little bit here. This is an extractor that sucks out the blood when you're bitten through the fangs. It doesn't really work. It just sucks. It doesn't work at all. These are pigs. We did an experimental study, and when we applied the suction versus the control where we didn't suck the venom out, there was no difference in the severity of the bite. In fact, some of the patients that we applied the suction had more damage caused apparently by the suction. Maybe it brought some venom, kept some venom locally, and it caused that degradation, that destruction of the tissue. This slide simply tells us that many drugs more derived from vipers than any other type of animal. We do get these amazing medical products from animals, including antivenom, that's used to treat bite. I can't tell you any more about, well, I'll tell you real quickly, I got a few minutes. So how do we get this antivenom? It's the only effective treatment for snake bite. We have to get the venom from the snake, and often we have to use venom from different snakes to produce these antibodies because the venoms often differ from species to species. We take that venom, we inject it into a horse or a sheep, does not harm that animal. And then over time, after a series of injections, we bleed those animals harmlessly, just like we get blood from you. We take that blood, we purify the proteins in it, we call antibodies. That's antivenom. It saves human lives. There are people, maybe you've read them, maybe you've seen them on TV, that claim that no human life has ever been saved because of research on animals. It's baloney. It's fake news. It's baloney, okay? Saves every year many thousands of lives in the world, okay? Here's a product FDA approved from Rattlesnake, used to treat severe chest pain, small heart attacks, some heart procedures. I could go through some others, but let me jump into this dog vaccine. Let me skip the dog vaccine. It probably doesn't work well because, I will describe it. So they take venom from the western diamond rattlesnake and inject a somewhat weakened form of it into the dogs. They make these antibodies. But here's a southern Pacific rattlesnake in Southern California. Those are the proteins, and there's no antibodies to those particular proteins. They're different from this species. So the vaccine doesn't work for all snakes, okay? Brain surgery. I've got a student working in a lab, John Zhang, where snake venom causes you to bleed. During surgery, bleeding and swelling, especially in brain surgery, is a big problem, okay? And so they want to ameliorate that. So what they do is they inject venom into these rats for 3 days in a row. Little amounts. It makes the rat bleed a little. The rat uses its own endogenous mechanisms to reduce the amount of bleeding. On the 4th day, they do surgery, and there's less bleeding. So maybe someday, when you have surgery, or a parent, or a grandparent, or a child, maybe they'll get some rattlesnake venom injected before surgery so they have a better outcome. It's pretty wild, huh? I'm going to skip that one because I don't want to keep you too long. In conclusion, rattlesnake venoms are complex, they're diverse, they're well adapted for predation and defense. They make decisions about how they use their venom, which is truly remarkable. They pose serious risk to us, so we have to treat them with respect, keep our distance, don't mess with them, especially if you're male, and have alcohol on board. The venom's vary in composition, and understanding that variation can be important for treatment, and research is going to be essential to our understanding of how dangerous these snakes are and how their venoms can benefit us, okay? I got to tell you real quick. Consider coming to Loma Linda University. We've got graduate programs in biology. I forgot about this, Dave, sorry. A couple more minutes. We have master's and Ph.D. programs. If you have not heard, in graduate school, generally, you're paying zero tuition, and you're getting a stipend of $10,000 to $25,000 a year. You may have to teach laboratories with students, but it's fabulous, okay? So we've got some terrific fossil and geology researchers that you could work under doing research. We've got biologists, like a marine biologist, Steve Dunbar, studies sea turtles, and Ricky Escobar, some environmental sciences, some weird guy there. And I'm happy to answer any questions you have about Loma Linda if you want to come up afterwards. And I want to just thank you guys for coming out tonight. Who hates rattlesnakes? Hey, come on. Not after that. It's okay, you can hate them. But they're cool. They're interesting. Thank you. All right. Any questions? I know some of you have to run. What do they use their eyes for? They do use them to see, and they have good vision. The infrared vision from the facial pits simply enhances that vision, and it works under conditions when their normal vision isn't available, like on a moonless night or down in a burrow.
Well, so I know some students got to go for like room stuff or whatever. If you can please stick around for a couple minutes, I'd appreciate that while questions are going on, but if you really got to take off, go ahead and slip out quietly.
Yeah, I'll forgive you. Yes, sir.